Hello everyone. In this video, we are going to do a general overview of blood. So blood is a connective tissue because it consists of formed elements that are suspended in an extracellular matrix, which is the plasma. The formed elements are, of course, the erythrocytes, which are red blood cells, the leukocytes, that are white blood cells, and the thrombocytes, that are platelets. I just want to make note here that a hematocrit is the percentage of erythrocytes. So starting with erythrocytes, which once again are red blood cells, you recognize these cells as biconcave discs that are anucleate, except in the case of frogs. But these cells are packed with a protein called hemoglobin, and it is hemoglobin that transports the oxygen from the lungs to the rest of the body. There's also an important note that the cell membrane of red blood cells is associated with a protein called spectrin, and it is this protein that enables the blood to squeeze through the small capillaries. Also, red blood cells are produced in the bone marrow. They're also stored and broken down in the spleen. Now, because we're talking about red blood cells, obviously we need to talk about blood types, which are directly related to red blood cells. Blood type is determined by the presence or absence of a particular antigen on the surface of the red blood cell. Antigens are, of course, substances that trigger an immune response. And these are located on, once again, the plasma membrane of your erythrocytes or your red blood cells. You are type B if you have the B antigen, you're type A if you have the A antigen, you're type AB if you have both the A and the B antigen, and you're type O if you have neither. There's also the presence or absence of what's called an Rh factor, and we'll get to that in a second. Now, I want to make note that whatever antigen you have on the surface of your blood cell, you have the anti of that antigen antibody in the plasma. So what I mean by that is if you have the B antigen, you have the anti-A antibody in the plasma. If you have the A antigen, you have the antibody or the anti-B antibody in the plasma, excuse me. If you have both the A and B antigen, you have neither the anti-A or anti-B antibodies in the plasma. But if you have O, that means that you have both the anti-A and anti-B antibodies in the plasma. And that is an important note for when we get to transfusion. So on the note of transfusion, I just want to make note that the donation of blood is not necessarily the same as the donation of plasma. For instance, the universal blood donor is type O negative. The universal plasma donor is type AB. And so here is why. Type O, right, has neither A nor B antigens. Type AB has neither anti-A or anti-B antibodies. And so if someone is type A and they're given blood from type O, there's no risk of agglutination. And agglutination is just the clumping of cells when the antibody reacts incorrectly with the transfused blood group, right? And you don't want that to take place. So type O is considered the universal donor because there's no risk of that occurring because type O has neither antigen. The universal plasma donor, being AB, is the universal plasma donor for the same reason. It's just that it has neither anti-A or anti-B antibodies in the plasma. So once again, you run no risk of agglutination occurring. Now, coming to the Rh factor, which is what determines that type O negative is the universal donor. A person with Rh plus, we generally want to transfuse with someone else that is Rh plus. And that is because a person with Rh plus will not make anti-Rh antibodies. Now, a person with Rh minus will make anti-Rh antibodies. Thus, an Rh person can receive blood from both someone who is Rh plus and Rh negative, but an Rh negative person can only receive blood from someone who is Rh negative. And so this is why type O negative is the universal blood donor. So moving on to leukocytes. Leukocytes are your white blood cells that are highly associated with the immune response. You have two types of leukocytes. You have the granulocytes, which just means that they have the presence of granules. These are your neutrophils, your eosinophils, and your basophils. And that picture on the lower bottom, the lower right, that is a neutrophil. And you can see the four lobes. An agranulocyte just means that there's the absence of granules. These are your lymphocytes or your monocytes, and the picture on the top right is the lymphocyte. Starting with the granulocytes, neutrophils are distinguished by a nucleus with 3 or more lobes. The granules also do not stain. You can see the unstained granules in this picture on the top right. Now neutrophils, they function in phagocytosis. In contrast, eosinophils, such as in the bottom right, an eosinophil has a nucleus with 2 lobes, and those granules stain red. Now, an eosinophil is involved in responses to parasitic infections and, of course, allergic reactions. Basophils have a nucleus that is somewhat S-shaped. The granules stain purple. And what's interesting about the basophils is that they migrate via diapedesis, which is how they pass through the vessel walls, and they form these mast cells that are involved in the inflammatory response. Agranulocytes, once again, do not have granules. These are your lymphocytes and your monocytes. Your lymphocyte is spherical shaped, like very well spherically shaped. And you have two types. You have the B lymphocyte that's involved in the production of antibodies, and you have the T lymphocytes that are involved in direct cellular attack. Monocytes, unlike the lymphocytes, are generally U-shaped. And they also, like the basophils, migrate via diapedesis. And these guys form macrophages that are just directly involved in just attacking these pathogens that enter your body. And of course, last but not least, there are the thrombocytes, which are just these platelets that are involved in coagulation, so they're generally called clotting factors. Unlike your red blood cells, these guys are multinucleated. And so that is it for blood.